Hello everyone, I'm Dr. Raman. In our last practical class, we had covered the six-linked inheritance and other numericals related to that. And today we will just move forward with our exercise number 7, that is linkage and crossing over. Okay. Um, like our last lectures, we'll just cover the theory portion first, um, and then we'll do the numericals based on this particular topic. Okay. After that, we'll just do the second exercise, exercise number 8. That is the chromosomal origin mapping. Okay, these two topics are linked with each other, so I'll just make a aggregate lecture on both of these topics. Okay. Firstly, what is linkage? When two or more genes reside on a same chromosome, they are said to be linked. Okay. They may be linked together on the autosomes or connected through the sex chromosomes. The genes on different chromosomes are distributed into the gametes independently of one another. The genes on the same chromosomes, however, tend to stay together during the formation of gametes. This result in thus the result of test crossing dihaploid will yield different results depending upon uh, whether the gene is linked or on the different chromosomes. Okay. In this particular paragraph, there are two things. Let me get this done. Okay. Firstly, let you have these two chromosomes A and B. Okay. This is just you can say this is a chromosome. Okay. If these genes are linked, okay, they'll just tend to make the gametes like, okay, and other gamete will be like this, because these two genes they are linked. No doubt they are on the same chromosome. Okay. As they are on the same chromosome, so they just tend to move in the next generation through the gametes like this only. As these two are on the different chromosomes, so they tend to just separate out. Okay. So this is the law of independent assortment. So according to that law, okay, they just separate individually, and in this way you can say that these are the linked genes, so they just move to the next progeny like this. Okay. Next is crossing over. Crossing over, it is the exchange of segments between the two non-sister chromatids of homologous chromosomes. It is done to be the crossing over, as like you had covered this in the theory portion. For example, this is one chromosome. Okay. And with this, if you just change the colors, so some portion of, for example, this is a black and this is a white. Okay, just this. So this, a small portion of black will come on this chromosome and rest this white, and the rest will be the black. Okay, in this way they just exchange some of the genetic materials from uh, each other. Okay, in this way you can say that, and this is the crossing over. Twice the crossover frequency in the cosmic frequency. The genotype AB will produce the following type of gametes. Okay, as I had already told, if you have B, A, B, okay, If there is linkage, then we will have only two kind of gametes. One is like this, another is A and B. Okay, because these both are linked with each other, so they just come into this form. Okay, this is complete linkage. Okay, complete linkage. Only these two kind of gametes. If the linkage is incomplete, so there are some chances of crossing over. Okay, if there are some chances of crossing overs, so we can have these recombinants. Okay, this is the parental, this is the parental, and these are the recombinants. So we can have recombinants, but as the linkage is incomplete, so there are the ratio will be like the parental population will be more than the recombinant population. Okay, if there is no linkage, okay, if there is no linkage, then these four gametes will be, again these four gametes will be formed. 1, 2, 3, 4. These two are the parental and these two are the uh, your recombinants, and the proportion is 50% of the parents and 50% of the recombinants. Okay, okay. Arrangement of the genes, the linkage between the two genes of an individual heterozygous for both pairs may be either of two types. On the basis of tendency of the genes to be inherited into the F2 generation, stay close or apart. The linkage is classified into mainly two forms. Okay, linkage is of two types. One is coupling, another is repulsive or repulsion. Okay, coupling term came from couple. Couple means when they tend to be together. Okay, repulsive means when they try to move away from each other. Okay, uh, coupling, the tendency of two dominant characters inherited from F1 from parents to stay together on in the F2. Okay, these are the okay, these are the dominant characters. So when we cross with the recessive, okay, this dominant will stay together like this, and this other character will stay like this. Okay, this is the coupling phase or coupling type of linkage or coupling type of link genes. Okay, this is also known as the cis arrangement. In the repulsion form, the tendency of two dominant characters inherited from one and another from the parent, another parent, tend to stay together apart into the F2 generations. For example, this is one kind, this is the another kind. So in this way, this will exchange each other with each other. Okay, this capital A will be replaced with small a, and small v will be replaced with the capital B. Okay, one will be the same. Okay, other will be the from the other parent. Okay, here the whole will be same. Okay, here they just changed. Okay, this is the repulsion, and this kind of repulsion is also known as trans arrangement. Okay, if R is the frequency of recombination, then the proportion of different gametes produced by a parent A, B, capital B by a small b will be under. Okay, if the coupling is there or the repulsion is there, okay, then coupling you can see coupling is cis arrangement or repulsion is the trans kind of arrangement. So if this kind of arrangements are there, then the proportion of gametes, or that is also known as the frequency. Okay, frequency of proportional gametes वो क्या रहेंगे? Okay, frequency और proportion क्या रहेंगे? This is the parental type. I'll write parental with P and recombinant with R. Okay, so this is the recombinant type. This is the recombinant type. Okay, this is a parental type. There is a printing error. Okay, here it is small and small p. Okay, so these are the parental forms. This is the recombinant forms. Okay, so in this particular case, if R is the frequency of recombination, okay, if the R is the frequency of recombination, the frequency or proportion of the gametes is for the parental form with a capital, that is half bracket 1 by R, or you can write like 1 minus R by 2. Okay, this is the generalized formula for both the parental forms. This is the formula, and for the recombinant forms, in the case of coupling, it is R by 2 or half R. Okay, if you just check the repulsion form, and then this coupling and repulsion, they are completely opposite. Okay, if you talk about the parental, then it is half R, and if it is the recombinant, it is 1 by R divided by 2. Okay, so these are the proportion of gametes. The significance of the linkage, the phenomena of linkage has one of the greatest significance for the living organism, that it reduces the possibility of variability in the gametes unless crossing over occurs. Okay, the frequency of crossing over is of great use in construction genetic maps of chromosomes. If provided, if it provides great evidence for linear arrangement of the linked genes in the chromosome. And the fourth is, it increases the frequency of the genetical variations, which are raw material for the organic evolution. Okay, so in this way, this linkage can work, and this is the main rule of this linkage. Okay, uh, before solving these numericals, you just have to remember that if yes, we have a space, if there is no linkage, okay, if there is no linkage, then proportion of P is equal to proportion of R. Okay, in the case of no linkage. If the linkage is complete, C is complete. Okay, then 100% population will be of parental forms. Recombinants will be 0. Okay, if the linkage is incomplete, IC. Okay, in this particular situation, the proportion of parents will be higher than the recombinants. Okay, these are the three major rules. Okay, just Okay, these are the three major rules which you have to remember. In the case of no linkage, parental and recombinants will be equal. Okay, uh, the gametes, okay, uh, and the proportion of the new generation. If the gametes are more, then obviously that the chances will be equal. Okay, in the case of complete, the 100% will be the parental forms, and this recombinant form will be zero. In the case of incomplete, the parental form will be more than the recombinant forms. Okay. Start with first numerical. Consider an example of test crossing of dihybrid individual capital P, small p, capital B, small b with small b, small p, small b, small b. Okay. That is the poled black, that is the horned red. The type and frequency of the gamete produced by the dihybrid parent. Okay, if you just uh, check the Mendelian ratios or Mendelian crosses, dihybrid crosses, so we can easily say that this PP and BB. Okay, this is 2N. Okay, that is the number of gametes कितने होने चाहिए. We have two heterozygotes, so it is total 4 gametes. Total we have 4 kind of gametes. That is capital P, capital B, capital P, small p, capital B, and small p and small b. Okay, this is from this parent, and from this parent we have small p, small b, and small b. Okay, because they all are recessive in the homozygous condition. This is the only kind of genes, gametes they gonna produce. Okay, now this is the if we are crossing with the what do you say, recessive homozygous. Okay, this is your recessive homozygous. If we are crossing one individual with the recessive homozygous, then this is the test crossing. Okay, so in the case of test crossing, we are testing that the genotype of the individual. Okay, so these are the 4 kind of gametes produced. Okay, and these are the single kind of gametes produced by this particular individual. Okay, the types and frequency of the gametes produced by the dihybrid parents. Okay, this is the types of gametes they produce. If there is no linkage, if there is complete linkage. If there is no linkage, in the case of no linkage, I had told you the parental form will be equal to the recombinant form. Okay, so 1, 2, 3, 4, these 4 kind of gametes will be produced in equal proportion. Okay, in the case of no linkage. Okay, so your first point is, in the case of no linkage, these all kind of gametes are formed. Okay, will be found from this particular individual, and this single kind of gametes will be found from this particular individual. Okay, and in the case of complete linkage, I had told you that 100% will be the parental forms. Okay, that is 100%, and the recombinant will be 0. Okay, so parental forms are this capital P, small b. Okay, this is the dominant one, and this other one is small p, small b. Okay, this is the another one. So these are the kind of gametes which this particular individual will form in the case of complete linkage. Okay, and in the case of this particular individual, this similar gametes will be found in the case of no linkage or complete linkage. Okay, so this is all with this first particular question. Okay, what will be the gametic output of capital A small a, capital B small b, and capital A small a, small a, small b, when crossing over occurs between the two loci and draw different inferences. Okay. Similarly, like this, okay, and this individual will form gametes like this. Okay. If they are saying that crossing over occurs between the two different loci, okay, so now the crossing over is there. We are not knowing that it is no kind of no linkage kind of situation, or incomplete kind of situation, linkage kind of situation. So in this particular form, question will have similarly two kind of commits. Okay. Small b, small a, capital B, and small a and small b. Okay, these are the four gametes which are formed from this individual, and from this individual will have small a and small b. Okay, as we are not knowing that which kind of situation is there in this particular uh, animal or this particular individual, so the generalized inferences, either in the case of incomplete or no linkage, we will have these four kind of gametes. If it is an incomplete, then this and this, they are the recombinant forms. So the recombinant forms will be less, and this are the parental forms. They will be more. Okay, very simple. Okay, we'll move further. Assume that the gene A and B are linked and is shown 40% of the recombination. If plus individual is crossed with one of the AB. What will be the genotype of F1? What gametes will F1 produce, and in what proportion the F1 is crossed with the double recessive? What will be the appearance of the genotype of the offsprings? Okay. Now we'll just do this particular question. Now from the very first line, if we see that the recombination percentage is 40%, means R is equal to 40%. If recombination is 40%, then what will be the parental form? That is 100% minus 40%. Okay. In a 100% population, 40% if the recombinants are there, then rest we have 60%, which are the parental forms. Okay. So from this data, if parental form is higher than the recombinant form, then this is the situation of incomplete linkage, where parental form is more than the recombinant form. Okay, so from this first line, we can have this inference that parental form is more than the recombinant form, so it is a kind of incomplete kind of linkage. Okay, of the recombination of plus individual is crossed with the AB. Okay, so in this particular situation, if what gametes will be produced when if we are crossing. This plus plus shows the dominant form, and this AB shows the recessive form, okay, of the particular gene. So if plus plus by plus plus, it is crossed with AB, AB, okay, this will form AB kind of gametes. This will form the plus plus kind of gametes. So the F1 will be like plus plus AB, okay. This kind of F1 will have, okay. So if we are having this kind of F1, okay, and we know that the recombination is 40% and parental is the 60%, okay. So if the combinations, if we are checking the combinations, combinations are like this. uh, Something on the screen, yes, okay. Firstly we have this plus plus kind of gametes. Other is AB kind of gamete. Third is plus A and plus B. So total we have four kind of gametes. Okay. If the recombinants are 40%, means this plus this is equal to 40%. Okay. And the parental forms are 60%. So this plus this, they form the 60%. Okay. So as we know that the ratios are always equal, okay, there are equal chances of gametes to be formed. So we can say that this plus plus will be 30%, this AB will be 30%, they both aggregate will form the 60%. Similarly, plus A will be the 20%, plus B will be the 20%, and their aggregate is 40%. Okay, so from this kind of quotient, firstly you have to check at which kind of linkage is there. This is the incomplete kind of linkage. So this is the kind of uh, what you say percentages we can have. Okay, but if we say that uh, we are there is no linkage, if just an example, if there is no linkage, okay, in the case of no linkage, what will be the gametes? Just apart from this portion, I'm just you that if the linkage is no, okay, there is no linkage. So in this particular situation, P will be equal to the R. So if P is equal to R, then these all have chances of 25% gametes. Okay, gametes will be having 25% chances. If there is complete linkage, then only parental forms will be there. Okay, these will have 0% and these two have 50% each chances. Okay, so if you're crossing it with the double recessive, so with that particular kind of quotient, with the incomplete linkage, we know the proportions are 30%, 30%, 20%, 20%. So if you're crossing it with a single this AB, Okay, so what proportion of individual we are getting? So we can easily calculate that. Okay. Okay. We'll move further. If individual homozygous for genes CD, this page is bit blurred, is crossed with the wild type. Okay, and the F1 is crossed back with the double recessive. The appearance of offspring is as follows. Okay, we'll having total 903 plus plus, okay, 897 CD, 98 D plus, and 102, that is C plus. Okay, this example is just similar to the example which we had already covered. Okay, so from that we can say that this is a parental form, this is a parental form, these two are the recombinant forms, these two are the parental forms. So as you are seeing that the parental forms are more than the recombinant forms, so it is also a kind of incomplete linkage. Okay, each and every time you just firstly see that which kind of linkage is there. Okay, you have to write that as the number of the recombinants are less, so it is a kind of incomplete kind of linkage. So in this particular kind of incomplete linkage, now we'll see what we have to calculate. Okay, the appearance of the offspring is as follows. Explain this result giving the strength of the linkage between C and D at um, assortment between C and D were independent. What would be the result of this cross? Okay. Um, in this, the major thing is we have to calculate the percentage of recombinants. Okay, percentage of percentage of recombinants. Okay, this is also known as crossover value. I know this writing is very poor. I'm just trying to write, but you just have to bear with me in this particular situation. Um, Also, it is known as a recombination frequency, known as the distance between the genes. Okay, so how we are calculating that? It is that the recombinants are this, these are the recombinants. So the formula is number of recombinants divided by total offsprings into 100. Okay, that will give us the percentage of recombination, okay, or the crossover value. So the kind of what we can get from this kind of table, that this is a kind of incomplete kind of linkage with the percentage of recombination that is equal to this is 98 plus 102. These are the number of recombinants divided by total, that is 903 plus 897 plus 98 plus 102. Okay, so multiply by 100. That will give us the 200 by 2000 into 100. Okay, this, this, this. So overall, that will give us the 10. So percentage recombination, or also which is known as distance between the genes, so that will give us the value of 10 map units. Okay, so as in the last line, what would be the result of this cross? What would be the result of this cross? This is a kind of incomplete kind of linkage where the percentage of recombination is 10 map units, or the crossover value is 10. Okay, so this is all with this particular question. Okay, next is the gene chromosome mapping. Gene mapping or chromosomal mapping in this particular kind of topic, the important point is 2-point test cross or 3-point test cross. Okay, this is very important on the basis of the numericals. Most of the competitive exams, they are asking the numericals from this particular calculating the chromosomal mapping and the gene mapping questions. Okay, so the gene mapping, it is a probability of the crossing over occurring between any two particular genes increasing as the distance between the genes become larger. Therefore, the percentage of crossing over or the combination frequency is used as a criteria for determining the distance between the genes. The map unit measures the distance between the linked genes. Map unit, that is 1% crossover recombination. Okay. Um, I think this, I had already told you that the distance between genes, it is we are calculating in the form of map units, also known as crossing over percentage, or crossover value, or percentage recombination, or the recombination frequency. Okay, so the map distance, if in this line there is a point that if the distance between two genes is more, there are chances that they will just do the crossing over. Okay, crossing over is more. But if they are closely linked, closely present, so there are chances that linkage is more. Okay, always keep this in mind. If two genes are far apart from each other, or if uh, they're present on the same chromosome, then there's present on same or on the different chromosome, there are chances that they will just do the crossing over as distance is more. If the distance is very less, or you can say that the distance is less and they tend to just move together, then the linkage is more. Okay, or linkage is there. Map units, it is okay. The map distance, the gene are present on the chromosome in a linear fashion. Linear fashion, I had already drawn a diagram that on a single line we have A and B. Okay, in this form, the from on a single chromosome, these genes present in the linear fashion. Therefore, it is necessary to find out the gene order and the gene distance. Okay, if we have a chromosome that has A, B, C, three genes. Okay, A, B, C. This chromosome has A, B, C, three genes. This also has A, B, C, three genes. This also has ABC, three genes. Okay, so we don't know what is the gene order, like how they're present. Okay, so for that we have to calculate the distance. We calculate the distance among each individual genes. So from that we can just say that yes, this kind of arrangement is there. Okay, with respect to each other. Similarly, the gene distance, distance I had already told, if the distance is more, then there are chances that the crossing over is more. If the distance is very less, then there are chances that these genes are kind of linked genes. So the gene distance and gene order is very important. Okay. Map distance, it is measured in the term of map units or centimorgans, which is also known as cm. One mu is equal to the 1% crossing over the recombination frequency. Recombination frequency, it is the number of the recombinant types divided by total number. Okay. If you have to calculate the percentage, then we'll multiply this with the 100. Okay, so that will give you the uh, recombination frequency. Okay, 2-point test cross. 2-point test cross, it is a very easy way to detect crossing over gametes in the dihybrid form. Okay, through the test cross progeny, we'll just I'm sorry, uh, from the 2-point test cross, we can easily calculate the crossing over between two characters. Okay, if the characters are more, then it is if the characters are three, then it is the 3-point test cross. Okay, 3-point test cross, it is used to determine the loci of three genes in an organism. Okay, if we have to check the location of two genes, then we use the 2-point test cross. If we have three loci which we just have which we have to test, then we go with the 3-point test cross. Okay, 3-point test cross, 2-point test cross, it is just you can say that a deviation from the Mendelian dihybrid cross. Okay, in the Mendelian dihybrid cross, we say that if we are doing um, test crossing of the hybrid. Okay, for example, this is the hybrid for PP, PB. Okay, this is the hybrid, and if we are doing the test crossing, sorry, this is P, P, B, B. Okay, so this is the test cross. So that will form the P, B, and this will give four kind of genes, four kind of gametes. P and small b. Okay, these are the four kind of gametes. So with that, we'll have four kind of individual with 1:1:1:1 ratio. Okay, in the F2 generation after the test crossing. But in with the crossing over and all, this value changes from 1:1:1:1. So this is just kind of deviation. Okay, that means if deviation is there, then there are chances that the linkage is there. Okay, so in this way we can just calculate the recombinants. If the recombinants are more or parental forms are more, if parental forms are more, recombinants are less, and there are chances that it is kind of incomplete kind of linkage. If the recombinants are equivalent to the parental forms, then we can say that this kind of linkage is the no linkage kind of situation, because it is 1:1:1:1. That is the normal Mendelian ratio. Uh, If we are just going with only parental forms, if we are getting only parental forms, then there are chances that it is a kind of complete linkage. So from that only we can just see that which kind of linkage is there. So if linkage is there, then what is the recombination percentages? So with this is all in this 2-point test process. Okay. In the 3-point test cross, the double crossovers usually do not occur between the genes less than 5 map units. For genes apart, it is advised to use third marker between two in order to detect the double crossovers. Okay, for that, I have to go to another slide. I'll share one more. Okay. 3-point test rows, and this is the part of your theory that just for your information. I have added this into your class. So these are the three genes. So if they all come into the parental forms and to the gametes, then it is a parental form. Then there is no crossing over. So these are the linked genes. Okay. If they are having crossover on one point, then it is a single crossover. In the last slide, it was showing that double crossovers and single crossovers. So just in order to give you a brief about the single crossovers and double crossovers, this is the so if on a single point we are having a crossing over, and other point there is no crossing over, this is a single point as crossing over. So if A and B are crossing over with each other, there is no crossing over in the C. So the C will be will remain same. Okay, uh, that means A or B are showing crossing over, means they are non-linked genes, and B and C are the linked genes. So if there is crossing over seen, this BC will go up, this BC will go down. Similarly, you can see, and this first uh, this A B, this BC will come up, this BC will go down. Okay, so this is the single type of single crossover. Now, if the crossover is written between the P and C, so this AB is linked, this AB is linked. So at this point, this will go down, or this will stay up, C will come up, and this will stay here, and C will come down. So this is a kind of single crossover. In the case of double crossover, you can say that no one is linked with each other. Every gene is free to move. So if A will be and B will exchange with each other, BC will exchange with each other. So this B will come up, this B will come down. So they'll make a double crossover. Okay. So if we are having this kind of phenotypes, okay, this is the parent type, parental type, capital and smalls. Okay, these all are the recombinants. Okay, now we have parental forms. So we have this parental form, and this is the recombinant form. So you can easily see that this parental form is um, uh, less than this recombinant form. Recombinant form is more. So we can easily say that there is a kind of crossing over. Okay, if crossing over is there, then only we are having these recombinants. Okay, if crossing over it is between A and B. Okay, if it is between A and B, then we can see that A and B are crossing over with each other, but B and C, they are linked. Okay, here B and C are linked. If B and C, then A and B are linked. Okay, A and B are linked. You can easily see. Okay, here B and C are linked. Here A and B are linked. Okay, if it is between A and C, then this is the double crossover kind of situation. Okay, so if we have to calculate between the recombination frequency, so we'll just have these assumed frequencies. So this is the total, so we can easily calculate the recombination frequencies from this particular data. Okay, now we'll go back to that slide, that particular topic. Yes. So the another topic, another portion is square root method. One is from the that particular data we can calculate, as I had already told in the last numerical. Okay, we can calculate the map units distance. Similarly, there is another method in the coupling phase. If the F1 parents are in the coupling phase, the frequency of the parental gametes, that is 2 square root of r. 2 square root of r. R is the recombination percentage or recombination frequency. Okay. The frequency of crossovers, if it is in the repulsion phase, then it is 2 square root of r, where r is the frequency of the double recessive. Okay. Now we have frequency. Assume the genotype ABAB is test crossed to produce two F2 consisting of this, and this. Estimate the percentage of recombination when the A and B by the square root method. Okay. Um, the recombination method, R is the frequency of double recessive. R is this. Okay. This is the frequency, that is 4. 4 that is 2 multiplied by the square root of 2, that is equal to the 4. So the recombination percentage by the square root method will be the 4. In study of autosomal linked genes, we test crossed dihybrid individual in the coupling phase AC, small a, small c, was found that the 37% of dominants at both loci, 37% recessive at both loci, 13% were the dominant at one locus and recessive at second, 13% were the dominant on second and recessive on the first. The similar kind of situation. This is the dominant on both, dominant on one, recessive on second, dominant recessive on second, first, dominant on second, both recessive. Okay, so this kind of situation. So we have total 13 recombinants, 13 recombinants. So total we have 26. Okay, recombinants, and total is 37 plus 37 plus 13 plus 13. Okay, that is equal to 100. Okay, so the how many map units distance they are apart from these loci. So that is, the formula is the recombinants by total into 100. Okay, that will give us the recombination frequency. So that is 26 by 100 into 100. This will cut with each other. Then the answer is 26 centimorgan or 26 map units. Okay, so these two, A and C, they are like this, A and C, with the distance between them is 26 centimeters. Hmm. These numericals are very easy. You just have to just memorize the formula. The reading is all based on formulas. If you are just getting the formula, and everything is easy. Okay. Um, from the test cross, two loci A and B, 24 out of 20% is well examined to be recombinants. AB, if you're talking about AB genes, the recombinants are 24 out of 200. Okay. If you're talking about B and C, then that is 12 out of 200. Okay. So we have to calculate the combination frequency. This will cut with each other. This is 12. This will be the 6. So A and B will have 12 map unit distance. And B and C has 6 map unit distance. Okay. Uh, Determine the distance between three loci. We had these two datas. So we had already calculated. Can the order of the genes can be determined from this information? Okay. So if you have to locate the genes, then we always have to follow this. Always pick the largest distance first, A and B. Okay. This is the 12. Okay. Now other is C. Okay. C is 6. So we don't know that C is on this side or it is on the this side. Okay. C. Okay. This is not clear because this recombination data for A and C we are not having with us. We have data for A and B, we have data for B and C. If we will have a data of A and C, then we can easily calculate the distance between A and C. So we can say if the distance between A and C is 6, if it is 6, then the pattern is A, C, B. This color is not visible. Yes. A, C, and B. Okay. If we are having the data of A and C, and the data suggests that it is 6 centimorgan. Okay. If we are having data of A and C and it suggests that the distance between A and C is 12 plus 6, that is 18 centimorgan. So in that particular situation, A, B, and C, this will be the loci of A, B, C genes. Okay, so can the order of genes be determined from this information? So you have to say no, because we are not having information between A and C. There are two combinations. Okay, either it is A, C, B, if the distance between A and C is 6, or it is A, B, C, if the distance between two genes is 18 centimeters. Okay, so I think this is clear to all. So Thank you so much. This is all about today's lecture. I hope you are getting each and everything. After completion of all the practicals, I'll just share the practical file into the group. So after that, you can just get the prints and you can do the numericals. Okay. Thank you so much, and have a good day.